You and your siblings inherited your DNA from the exact same two parents, yet you likely display entirely distinct physical traits. This happens because of a microscopic mechanism called crossing over. During meiosis, as germ cells are produced, homologous chromosomes physically entangle and swap matching segments of DNA before being passed to the next generation. Without this precise exchange, sexual reproduction would yield identical genetic combinations in every gamete, eliminating the vast diversity we see across populations. Here is a bivalent diagram. Crossing over is defined strictly as exchanging genetic material between non-sister chromatids of homologous chromosomes, one maternal and one paternal. This exchange is facilitated by the synaptonemal complex during the Pachytene stage of prophase 1. The process involves 3 physical steps. First, homologous chromosomes align. Next, chromatid segments break and cross at a point called the chiasma. Finally, they reattach to form brand new recombinant chromatids. For the WBCHSSE board exams, using the terms non-sister chromatids and Pachytene stage is essential for securing full marks on this definition. We classify crossing over by its source. Germinal crossing over occurs in meiotic cells during gamete formation and is universal in sexually reproducing organisms. Somatic crossing over occurs during mitosis in somatic body cells. It is exceptionally rare and has no major evolutionary impact. We also categorize these events by the number of chiasmata formed. A single crossing over event involves breakage and reunion at only one point in the tetrad, yielding 2 recombinant chromatids. If the chromatids cross at 2 different points, it is a double crossing over. If they cross at 3 or more points simultaneously, it is multiple crossing over, though this is statistically very rare. While single and double crossovers drive variation seen in offspring, multiple crossovers occur so infrequently that they are rarely observed in a standard population sample. Genetic linkage is the tendency of genes located close together on the same chromosome to be inherited together. Because they are physically near each other, they are less likely to be separated during meiosis. This creates an inverse rule. As the physical distance between 2 genes increases, the chance of a crossover occurring between them also increases, which causes the strength of their linkage to fall. Because crossing over breaks these links, we use recombination frequency as a direct proxy to calculate the physical distance between genes on a chromosome. To calculate that distance, we use the recombination frequency formula. It is the number of recombinant offspring divided by the total number of offspring multiplied by 100. This percentage translates directly into map units. A 1% recombination frequency equals 1 centimorgan. So, if 2 genes show 15% recombination, they are 15 map units apart. Let's look at an exam problem. We have 4 genes with these crossover frequencies. A to B is 10%, A to C is 6, B to C is 4, A to D is 3, and C to D is 9%. To start your map, always identify the largest percentage first to establish your anchor points. Since A and B are 10 units apart, we place them at the ends of our map. Pause the video here. Using all the frequencies provided, try to determine exactly where gene C and gene D fit on this line. Let's solve for gene C first. It is 6 units from A and 4 units from B. Because 6 plus 4 equals our total span of 10, gene C must sit directly between them. Now for gene D. We know it is 3 units from A. This allows for 2 possibilities. D could be 3 units to the right of A, inside the segment, or 3 units to the left, outside the segment. We resolve this using our final data point. The distance from C to D is 9 units. If D were inside the segment, it would only be 3 units from C. Because it is 9, D must be positioned to the far left. The final correct sequence is D, A, C, B. Mapping is simply a process of deductive reasoning, testing relative distances until they all align on a single linear path. Use this checklist for your final review. Remember, crossing over occurs between non-sister chromatids during pachytene. It is the inverse of linkage, and 1% recombination equals 1 centimorgan. Mastering these calculations allows you to transform abstract inheritance patterns into a physical, measurable coordinate system for an organism's genome.